Thank you very much. Thank you very much. It's a privilege to be here, and I must say I was quite surprised when Christy rang me up and asked me to give this presentation. I hadn't considered that I would be asked, so I was very honoured, and thank you for having me tonight. As you can tell from the title, I'd like to talk broadly about challenges that we face in soil science in terms of food production. But I want to talk globally, but I also want to talk at a very fine scale as well. And hopefully the reasons why I want to make these comparisons will become apparent as we move through this presentation. I'll just start with some facts and figures to introduce, which I'm sure you all know, but will be helpful in setting the scene. We all know that the world population is increasing. And depending on projections, by the year 2050, we expect about 9 billion people on the planet. Okay, and so that's a huge increase in people. And what's particularly relevant for us as soil scientists is that 97.6% of the calories that we consume as humans come from soil. Okay, so the remaining 2.4% come from aquatic ecosystems. 97.6% of our food comes from soil. And so these extra humans, which we're going to have in the world, are going to have to be fed from the soil. So how are we actually going to do this? Well, it's not going to come about because we've got double the land area to crop. Okay, this is from the FAO Agricultural Outlook from this year. And so this is just some selected data showing the average annual cropland change for selected countries. And the dark blue is 2007 to 2016. And then the grey is 2017 to 2026. And so you can see that in some places we're going to have more land, but in other places we're going to have less land. And in fact, when you start to look at individual crops, so this is for maize, and on the left-hand side you can see the area expansion, which at best is extremely modest. Okay, so the area expansion, you're looking at 5–10% increase in the area which is going to be cropped. Rather, the increase in food production, which we need to feed these extra billions of people, is going to have to come from increases in yield. So by the year 2050, we're going to have to grow twice as much food from the same amount of land. Okay, and so as soil scientists, this is our challenge. How are we actually going to achieve this? How are we going to, in amongst with all the other opportunities, all the other challenges, all the other disciplines, how are we as soil scientists going to ensure that we can actually achieve this? So on one hand, we need to double food production per unit of land. On the other hand, we have a whole range of environmental challenges which we're already facing. And so I'm sure many of you will have seen this. This is from 2005. It's the planetary boundaries concept. And so in many areas, many of which are related to soils, as you glance around this diagram, you'll see several of these are interconnected with soils. We're already exceeding planetary boundaries in several of these areas. Nitrogen and phosphorus stand out in particular. You'll see climate change there. We'll talk a little bit about climate change this evening and the role of soils in climate change. Land system change, loss of genetic diversity. So on one hand, we need to double food production from the land that we already have, but on the other hand, we're already exceeding the planetary boundaries, many of which are related to soils. So how are we actually going to do this? This is an image which some of you may have seen me use before. This is from China, from the Loess Plateau. And you can see not only do we have these global planetary boundaries, these global challenges, but if we now start to hone in on soils, how are we going to double food production from the same land area when this is some of the problems that we're experiencing? And I'm not sure if you can pick them, but there's two people standing there which give a bit of scale to that image. Unfortunately, the top little bit's chopped off. So soils provide fertility they filter and store our water, they provide structural support for plants, sequester carbon, contribute to biodiversity, and they provide raw materials. In fact, depending on how you do your calculations, we know that soils provide us with goods and services equal to approximately $12 trillion per year, which is roughly equal to 15% of the world's GDP. So this soil resource, which is extremely valuable, $12 trillion per year in goods and services, we need to double production whilst maintaining that resource and remaining within the planetary boundaries. Okay, and this is the challenge that we face. So what I want to, within that framework, what I want to look at this evening, now I should point out that no one has ever actually told me how long I can speak for tonight. It was neglected in all the emails, so you got another 2 hours, so you're pretty comfortable. The question I want to ask tonight in the framework of that introduction is, if this is what we need to achieve, what are we already doing? Okay, what are we doing with our soil resource? What in our current agricultural systems, what changes do we see in our soil properties, and how, with these changes and our current systems, how are we going to double our production? So what changes in soil fertility does our long-term production of food cause, and how does that fit within the challenges that we have going forwards? Now, there's a whole range of topics that you could talk about within this, okay? And clearly, I'm not going to cover everything. You could talk about salinity, you could talk about a whole range of things, okay? And I've picked off, 3 or 4 topics that I would like to look at. And obviously, I think they're important topics, but it's not an exhaustive list. So the first thing I'd like to talk about is carbon, okay? Which maybe at first seems like a bit of a boring element, carbon. But we know that carbon in soils is extremely important. The clearing of land for agricultural cropping depletes the soil organic carbon. Okay, and that has implications on several factors. One is its impacts on soil fertility. So this loss of soil organic matter, this soil carbon, decreases soil fertility because we know that both the content and the composition of the organic matter changes over time, and that there are changes in the chemical and biological processes occurring within the soil as a result of that loss of organic carbon. And I've got some data on that in just a moment. But the other one, and the one which many students at an undergraduate level don't immediately realise, is the role of soils in greenhouse gases, in global warming. And as I'm sure most people here would know, soils contain more carbon, more organic carbon, than is in the atmosphere and in the biotic pool combined. So we have this enormous reservoir of carbon within the soils. And what is the impact of, so my question is, what is the impact of our agricultural production on this pool and the contribution of carbon to the atmosphere as carbon dioxide as a greenhouse gas? So, at a global level, this is some of our data just published this year. And there's a whole range of things in here which I'm not going to go through in detail. The ones which are of particular interest are the ones in the red boxes, which is where you convert a native ecosystem, a native vegetative ecosystem, into a cropping system. And this is all long-term data that's presented here. So this is, to make it into this data set, it had to have been cropped for at least 15 years. Okay, so I'm only concerned about long-term at the moment. And so what you'll notice here is that where you convert native systems into a cropping system, after long term, you have lost roughly 40 to 50% of the carbon to the atmosphere as carbon dioxide. And so there's implications there for greenhouse gas emission, but also there are implications for soil fertility. That the organic matter, which is central to maintaining the fertility of the soil, you've lost half of it. Okay, there's a whole heap of data which goes into this, and if anyone would like more details, I'm happy to talk about it later. What's the depth of sampling, etc., etc. Okay, but across the world, this is a global meta-analysis. Across the world, in long-term systems, we lose 40 to 50% of the carbon in our soil to the atmosphere as carbon dioxide. And this is a map, and this is just published last year. This is not my data. But this is just a map of the world, and this is showing the acceleration in the turnover of soil organic matter, soil organic carbon. Okay, so this is the acceleration, and you can see from the legend there, you can see the sort of values you're talking about, that at its highest value, you're talking about a 20 times acceleration in the rate of organic matter turnover. And you'll note that the acceleration occurs largely within the areas of high productivity in terms of our agricultural production. Okay, and so here we have this situation where this long-term production of agricultural products is causing this acceleration in the turnover of organic matter, and it's lost to the atmosphere as carbon dioxide. And we know the reasons for that. I mean, we don't need to talk about that now. We know that you have increased soil temperatures, and so the microbes like that, and they have increased activity, increased oxygen, etc. So, what is actually going on here? Despite the importance of this, the mechanisms by which carbon is stabilised within soils remains unclear. So despite the importance of this, and despite people having worked on this for their entire careers, this is such a complex problem that there is just so much that we don't know still. Traditionally, it's been assumed that the stabilisation of organic carbon in soils is related to three properties. The first is the formation of these complex, highly complex, large compounds which are resistant to microbial degradation. The second is the formation of organo-mineral associations. In other words, the organic matter is being attached to the minerals within your soil and providing protection against its degradation. The third is that we have this physical, if you like, spatial inaccessibility of occluded organic carbon, which is inaccessible to microbial communities and enzymes. And so we have these three mechanisms, but we still don't actually really have a good handle on how organic matter is stabilised in soils, what sort of organic matter is going to be lost to the atmosphere as carbon dioxide, what's going to be retained, why. The current evidence is tending to suggest that the first point there is probably not as important as we once thought it was. Okay, that there's a lot of evidence that actually we're not forming these extremely large, complex compounds which are resistant to microbial degradation. It's probably more of the second two. And so now to bring this down, I guess, to my nano scale, if you like. So we have this global problem, but we can't actually understand what's going on in the soil. And this inability to understand the underlying processes hinders our ability to manage the system at a larger scale. And so I'm not going to talk about methodology in detail at all, but we took two soils from Queensland, which have been cultivated, one for 60 years and the other for 79 years. And we compared them. It was a paired site, so we had a soil immediately next to it which was undisturbed, and so had its original vegetation. And in these particular two soils, at 0 to 20 centimetres depth, we'd lost approximately 80% of the organic carbon in that cropping period had been lost to the atmosphere. And so what I want to very briefly show now is that we have these new approaches which were not possible 10 years ago, 20 years ago, where for the first time we can actually take soil, we can analyse it in situ without any preparation, and we can have a look at the distribution of carbon within soil microaggregates, and we can have a look at the forms of carbon within microaggregates, and we can start to do these in situ analyses. I'm sure that when some people started their careers, it was extracting soil organic carbon with all sorts of extractants and trying to make predictions about, well, this is what I've extracted, so what would it look like if it was originally in the soil? Okay, and we made some important observations, but we now have these opportunities to look at organic carbon actually within the soil in its current natural state, if you like, and see what's going on. Okay, and so as Bigsy was just teasing me 10 minutes ago, here you go, Bigsy, I managed to get the synchrotron in. So here's the Australian synchrotron down in Melbourne, okay, and I'm not going to talk about how synchrotrons work, other than to say it's the largest piece of scientific equipment in the southern hemisphere, okay, and the electrons zip around the storage ring There are magnets in the storage ring which accelerate the electrons and make them wiggle. Okay, and as you accelerate the electron, you generate a photon. And so we generate all sorts of different photons of different energies, from infrared right through to hard X-ray. And what's of interest to us today is these infrared. Okay, and so this is exactly the same as an infrared microscope that you might have in your lab here. In fact, this is a desktop conventional infrared microscope which they purchased commercially and then ripped out the guts of it, and they've plumbed in the infrared, the synchrotron photons coming in here. Okay, so this is exactly the same as you could do in the lab, except it's about 1,000 times more powerful. Okay, so you can have a look at carbon in the soils, which we haven't been able to do before because the organic carbon level in soils has been too low to use these sort of approaches. But we can now start to look at these things. And the heading is completely covered up in this one. So this is an undisturbed. Okay, I can't see the heading either. I'm pretty sure it's an undisturbed soil, and I think it's a vertisol. That might make it better. You can now half read it. So this is an undisturbed vertisol. I'm not going to go through this in detail, but what we actually did here was we took an intact microaggregate from a soil, undisturbed soil, we sectioned it with a diamond knife, so you cut through the aggregate, okay, cut through the centre of the aggregate, and you end up in the top left-hand corner. You can see what it looks like under a light microscope. Okay, and so you have this intact microaggregate sectioned to 100 nanometres thick, okay, which the Victorian government still needs assurance it doesn't contain any fire ants when you take it out. So you have this tiny little microaggregate, okay, and you can put it into the infrared microscope. And so what you get is you get a map showing the distribution of carbon, so the amount of carbon, but also the forms of carbon. And so in this little microaggregate, here we have the polysaccharide, carbon, aromatic, aliphatic, and then over on the far right-hand side we have plain minerals. Okay, and so what we can do is, first of all, we see that there's some nice relationships. So in these graphs here we have the relationship between the clay minerals, and on the y-axis we have polysaccharide carbon, aromatic carbon, and aliphatic carbon. And so we can see the relationship between clay minerals and the different types of organic compounds. Okay, and so we can see that in this undisturbed soil we have this really quite nice relationship between these different forms of carbon and the soil clay minerals themselves. So this tells us that in this soil there's probably quite strong evidence of organo-mineral associations, that the mineral framework of the soil is perhaps providing some protection to the organic materials themselves. If we now have a look at the crop soil, so this soil's been cropped for 70 years or thereabouts, and again, here's our microaggregate. If we just skip down to these graphs here, we can see something interesting. Okay, and that is that for the aromatic carbon, we no longer have a relationship between the soil mineral phase and the aromatic carbon. In fact, a lot of the aromatic carbon is gone. Okay, for the polysaccharide carbon, again, we see a lot of the polysaccharide carbon is gone, which isn't surprising when we consider what sort of carbon it is. Okay, but we still see quite a good relationship for the aliphatic carbon, and a lot of the aliphatic carbon is still there. So organo-mineral associations are of critical importance to stabilisation of carbon in soils, particularly for aliphatic carbon. I'm not going to, we have got other data on this as well, which just for time's sake I'm not going to go through. We said that mineral-associated carbon is not entirely resistant to degradation. But the other interesting thing is that our data do not, in this particular instance, support the hypothesis that the stability of soil and carbon in free stable microaggregates is regulated by spatial inaccessibility. And the reason I say that is that when you look at these diagrams at the top, the maps at the top, we can't see any apparent, whether it be for the undisturbed, for the cropped or the undisturbed, there's no apparent trend in the carbon concentration as we move through the aggregate. Okay, that if accessibility was the factor that was driving carbon degradation, you would expect to see a change in carbon forms and concentration as you move through a microaggregate, but we don't necessarily see that. Okay, so perhaps it's this organo-mineral associations which are important here. So to summarise soil organic carbon for the purpose of today, so it's important to identify the mechanisms which are influencing the persistence of organic carbon in soils. So if we are wanting to manage at a global level the fact that we see 50% of our carbon lost from soils, we see these trends, we see these changes, it's contributing to a decrease in soil fertility, it's contributing to greenhouse gas emissions at the same time that we're trying to increase productivity. To manage these systems, we need to know what's going on. And so for this management of soils worldwide, for modelling, etc., we need to understand the underlying processes. The second topic I'd like to talk about today is nitrogen. Nitrogen's another really interesting thing I find for the undergraduate students, because a lot of the students don't understand a lot of the history that goes behind nitrogen fertilisers and the linkages which are there. So nitrogen gas accounts for 78% of the atmosphere, but it's extremely stable. Okay, and because it's extremely stable, it's a triple bond, it takes a lot of energy to convert that into a nitrogen fertiliser. And so we know the Haber-Bosch process consumes something like 1 to 1.5% of all global energy around the world. And so we have this extremely energy-intensive process. And it's one of the really interesting things because the Haber-Bosch process is directly linked to the deaths of 100 to 150 million people through conflict around the world in the 20th century. But at the same time, we know that half of the world's population are directly reliant on the Haber-Bosch process to supply that nitrogen fertiliser to grow the crops to give them food. And so it's one of these really interesting things where the Haber-Bosch process, consumes enormous amounts of fossil fuels, has resulted in the deaths of 100 million people through armed conflicts because of its use for explosives. But at the same time, half of the world's population are reliant on it. But then actually by fertilising it, which we do not terribly efficiently, and a lot of the nitrogen doesn't end up in the plant, not only all of those things, but that excess nitrogen causes environmental degradation of our waterways, etc. With the Great Barrier Reef. So what about nitrogen? So again, back to the global level. This is, for those who know the soil chemistry well, would not be surprised that if you have a 50% reduction in soil organic carbon, you're going to have approximately a 50% decrease in your soil organic nitrogen as well. And so again, if we look at these graphs and we compare a native system to a crop system, we can see that 40–50% of our nitrogen has now been lost from our soils. I must say that when we were doing this study, Neil got very concerned with me because at the time I didn't have this final chart on the final bar on there. And Neil was concerned that the summary of my study was going to be that everyone should go to organic agriculture because organic agriculture is going to increase your carbon, it's going to increase your nitrogen. And he was very concerned. He called me into his office and said, Peter, this is not it. So I had to put in this final one, which is where you just apply an organic amendment. Okay, and so clearly it's nothing about organic agriculture itself, rather it's just the fact that you're putting organic compounds back into your soil, which is increasing your carbon or your nitrogen content. Okay, so nitrogen, we're losing 40–50% of the nitrogen from our soils in long-term cropping systems. What's actually going on? There's an interesting link here, and you'll see it shortly, between what we've just spoken about for carbon and for nitrogen. And so what we did in terms of nitrogen was Ram had these soils which had been incubated for 365 days with some pulse-labelled lucent residues, so carbon-13, nitrogen-15. And with some colleagues in Germany, we used a nanosynth, a nanoscale secondary iron mass spec. Okay, and because it's a mass spec, you can do isotopes, Okay, and so we decided to shove some soil in and see what we could see. This is a vertisol. Okay, and up in the top left-hand corner is a scanning electron micrograph. And so that's simply just showing you where the soil particles are. Okay, and so again, these are free microaggregates. These are not dispersed. Right, this is free intake microaggregates, very delicately removed in a suspension and placed onto this wafer. And so that's just a scanning electron micrograph, and so you can see the soil particles there. Down here in the bottom left-hand corner, this is, if you just ignore the different colours, blue through to purple, wherever you can see colours, whatever colour it happens to be, represents the organic matter. Okay, and so this is a new technique we have, a comparatively new technique, where you can actually visualise with about 50 nanometre resolution organic matter sitting on your soil particles. And so there's something interesting here, that the organic matter is not homogeneously distributed across the soil particles, rather it's in discrete patches on the soil. Okay, and so what's actually going on here? And I don't have an answer, but why is the organic matter attaching to that particular part of a soil particle and not another part? There are hypotheses out there. It's being proposed that, you know, is to do with rough patches, rough areas on your organic on the minerals where the organic matter is attaching, but we just don't know. Okay, so why is the organic matter attaching there? But then when we put in this radio-labelled lucerne residue and we incubate it for a year, we see something interesting. And so on the right here, this is nitrogen. Okay, and if we look at the colours here, this blue colour here represents, if you like, a native nitrogen. Okay, so it's nitrogen with the same isotopic ratio as was originally present. But wherever you can see the red colours or yellow colours, that's new nitrogen which is being attached to the soil. There's something really interesting going on here, that we see that the nitrogen, which is microbially derived residues, okay, so this is organic matter which has been chomped up by the microbes and consumed and is now very high in nitrogen. We see these nitrogen residues are attaching to soil particles where the carbon wasn't previously. So what's going on here? We see the carbon attaching to some parts of the soil but not other parts. But then we see these microbially derived residues attaching to new surfaces. Okay, they're not attaching to existing surfaces with existing organic matter, they're attaching to new surfaces. And so what we think is happening here, we see soil organic matter accumulating in discrete areas. Okay, so again emphasising the importance of these organo-mineral interactions, the fact that probably the soil mineral fraction is providing some protection, if you like, for the organic matter. But we see that microbially derived nitrogen, which is microbially derived organic matter, which is rich in nitrogen, is accumulating in localised areas of mineral surfaces not already co-localised with organic matter. And so this is what has been termed the onion model. In other words, we have these different sorts of organic matter, in this case microbially derived, rich in nitrogen, with different functional groups, which is able to attach to new areas of the soil and form these new surfaces, perhaps which will now in the future form other surfaces upon which more organic matter can bind. I don't know, okay, but that's a possibility. How to actually explain what's going on here? There's all sorts of hypotheses. This is the other soil, which is the curasol. We won't go through this in detail, except to say that there's some differences here when you compare it to the vertisol. So not only do we have differences between carbon, microbially derived organic matter rich in nitrogen, but we see differences in soils. Different soils behave differently. In this particular scenario, the vertisol, we know vertisols have highly reactive surfaces, etc., etc. Okay, and so perhaps not unsurprising that we do see differences between these soils. But as a soil scientist, there's a great opportunity here, isn't there? Because what's actually going on? What's actually going on? We can make these observations, but what do they mean? So we see the attachment of microbially derived compounds is less pronounced than for the vertisol. So to summarise organic nitrogen, in these soils, long-term cropping systems, we lose 40-50% of the nitrogen. That nitrogen is expensive to replace. Energetically expensive, economically expensive. we see that almost all the organic matter within the fine fraction of the soil was accumulating on mineral particles. Again, perhaps emphasising the importance of these organo-mineral interactions. We see that these nitrogen-rich microbial products originating from decomposition are absorbing directly to mineral surfaces. They're not attaching to where we already had organic matter on that mineral surface. And so perhaps these nitrogen-rich products are demonstrating the ability of soils to store organic matter in newly formed organo-mineral associations. So what sort of organic matter can bind to the soil, what sort can't, what are the differences between soils? Okay, we don't really have the answers to these questions yet. Very briefly, I'm not going to go into this at all in detail. This is phosphorus and sulphur. Okay, that we know surprisingly little about how phosphorus and sulphur changes soils over time in long-term cropping systems. Here we have phosphorus. Let's look at this one here. Phosphorus concentration in soils has gone down maybe 30%, 20-30% in these long-term systems. Here's sulphur, a similar amount, maybe 20-30% decrease in sulphur stocks within your soil upon long-term cropping. Another interesting thing you can look at here in this global meta-analysis is changes in ratios, carbon-nitrogen ratios in your organic matter. And on the right here we have carbon to sulphur ratio in your organic matter. And so what we see, interestingly, for nitrogen is that although we've lost 40 to 50% of the nitrogen from 40 to 50% of the carbon and nitrogen from our soil, we don't have a change in the carbon to nitrogen ratio of the organic matter. For sulphur, we do see quite a pronounced shift in our carbon-sulphur ratio within the organic matter. So what's actually going on here? Well, there must be differences in the mechanisms by which nitrogen and sulphur are mineralised from the organic matter, which again, perhaps is not surprising. We know, for example, biochemical mineralisation is important for sulphur, okay? But we can see this at a sort of global scale, if you like. Look, and the final thing I want to talk about today is soil acidity. So we see in these long-term production systems, we see a loss of carbon, a pronounced loss of carbon, loss of organic matter, loss of nitrogen, phosphorus and sulphur decreased by 30%. But there's a range of other things going on as well. And so I just want to finish talking about soil acidity. Soil acidity, you can see the map here, the red areas are acid soils, the blue areas are alkaline soils. And we know that about 3.95 billion hectares of the global ice-free land is acid, which corresponds approximately to 40% of the arable land that we currently have. The problem is that soil acidity is getting worse over time. Okay? That human activities, whether it be acid rain, whether it be agricultural production itself, are exacerbating the problem of soil acidity. And so soils are becoming more and more acid over time. And in these acid soils, we know that aluminium becomes toxic. So aluminium is the third most common element in the Earth's crust after silicon and oxygen. And in these acid soils, the aluminium becomes soluble and increases to levels which are toxic to plants. And in Australia, we know, or we estimate, that soil acidity costs us about $1.5 billion per year in lost productivity. Okay, and so again, if we think about our opening statement, which was we need to double food production from the same unit of area, we need to start to consider, well, how are we going to address some of these issues? We've known for over 100 years that aluminium is toxic to roots in acid soils. In 1904, it was found that the acid soils are high in aluminium, high in soluble aluminium, and we've known since at least 1914 that aluminium is toxic to plant roots. So despite that, in the 100-odd years since that time, we still really don't have much of an idea about how aluminium is toxic. Clearly we've made a lot of advances, but there's a lot we still don't know. And so to increase food production, it's necessary to understand aluminium toxicity. And so what I want to have here is, I've probably got 3 or 4 slides. It's a little bit of a shift from focusing purely on soil, and now I want to move to the soil-plant interface. Okay, and as you'll see on, I think it's my second last slide, I've only moved about 10 micrometers. Okay, I haven't actually moved very far at all. Okay. The first thing that really got, well, Pax, you're about to appear on here, but the first thing which really got me going on this was Pax had this observation that aluminium, copper and lanthanum cause similar symptoms in plant roots. Okay, and so here we have some scanning electron micrograph from Eugene Dalal, who did his PhD in the School of, what's now the School of Agriculture and Food Sciences, I guess it was the Department of Agriculture, I guess, at the time, with Colin Asher and with you, Pax? Colin Asher and David Frank Smith. Okay, and so here's this nice image from the scanning electron microscope. And interestingly, what we see in this is not a nice, happy root. We see a root which has got all these tears and ruptures in it. Okay, and so that root's got problems. And then here we have an image from 2001 showing aluminium. And you can see that that root with the aluminium has quite similar symptoms. The root is all ruptured and torn. And then this is from work from Anna, who I saw just before somewhere, trying to hide, with copper. I think from memory that might have been Rhodes graphs, Anna. And you can see with copper toxicity a similar sort of thing. So what's actually going on here? Here we have 3 elements from the periodic table, which if you were to pull it out, maybe you can see some similarities between the 3 of the elements. But why do we end up with such similar symptoms in completely different plant species? So here you are, Pax. So I'm quoting Pax as accurately as I can remember, but here's a hypothesis that we can test. Aluminium toxicity is attributed, so this is a decade old, right? So I'm trying to remember something from 10 years ago. Aluminium toxicity is attributed to a direct inhibition of entropic cell expansion in the elongation zone resulting from the strong binding of aluminium to the cell wall and prevention of wall loosening. Is that pretty accurate, Pax? Yeah. He's happy. Okay. So this is what we wanted to test. And so in typical style for me, and I don't know, maybe less so Pax, we've lost the top row of labels there, but we pulled out the periodic table and went nuts. All right, and so here you can see roots exposed to, I'm not sure what's in the top row, aluminium, other things I don't know, mercury, indium, ruthenium, scandium, copper must be at the top there, Pax, and something else, lanthanum. And the list went on. We tested all sorts of unusual things which we had trouble finding in the chemical catalogue. But interestingly, all of these things cause the roots to rupture. Okay, how can that be? And so, in fact, what we had done, Pax, if I remember correctly, we had actually sent, not this particular article, but we'd sent an earlier article off to review, and the reviewer had come back and said, hey, why don't you guys draw this sort of graph, and you might see a trend. And so we did draw the graph. Now, on this graph, please ignore silver, right? I'll talk about silver later if anyone's interested, but just ignore silver for the moment, it's a real pain. If you were to look at this graph, the things which in the black circles are things which cause the roots to rupture and tear. The things in the white circles are things which don't cause the roots to rupture and tear. And so you can see that all the things which are causing damage to the roots, causing this rupturing and tearing to the roots, are things which bind strongly to hard ligands. In other words, they're at the top of that graph. The things which don't cause the ruptures, with the exception of silver, are at the bottom of that graph. You know, silver is a problem. Okay, why is that? So what do we actually think was going on? So our hypothesis was, as Pax told us 2 slides ago, that things such as aluminium, which bind strongly to the hard ligands, including the cell wall, okay, so the cell wall in plants contains carboxylate groups, so in pectin, okay, and that aluminium is binding strongly to the cell wall. By binding strongly to the cell wall, it prevents the cell wall from elongating, And when you prevent the cell wall from elongating, and the middle of the root keeps on going because it's quite happy, it tears the outside of the root. Okay, and so that was the hypothesis, that this is a direct inhibition of growth caused by the aluminium binding very strongly to just the outside of the root, where it stops it growing and elongating, and so we end up with these toxic symptoms. Okay, so we set about testing this. I'm going to go through this fairly quickly, but here we have time, okay, and so we're operating on fairly time, but here we're dealing with, I think, 5-minute time intervals. And then here's our root elongation rate. And we added aluminium at time 0, where we have this solid vertical line. And so we can see at the high aluminium rate, 75 micromolar, that we've actually inhibited root elongation within 5 minutes. So faster than we could measure. In fact, what underpins this data and what's not here is we're actually taking measurements in 30-second intervals. So we're measuring how quickly roots are growing every 30 seconds, which you can actually do, to my surprise. It's a bit of a problem. Roots don't grow as you might immediately suspect. It seems roots sort of grow in circles and all sorts of things. So aluminium, toxic, and it's toxic within 5 minutes. The other thing we can do in 5-minute intervals is determine what is it that is actually reducing root growth. Okay, and so if you think about a root, and some of you probably know more about roots than others, if you think about a root, what's actually causing the root to grow is that we have this elongation zone. And I'm not going to talk in detail about this, but the speed with which a root is growing can depend upon how many cells are within that elongation zone, and hence how many cells are elongating at the same time. The more cells you have, the faster the root will elongate. Okay, but it also depends on the speed at which the individual cells themselves are elongating. Okay, so do you see the two factors? How many cells are in there and elongating, and how fast is each individual cell elongating? Gives you the overall elongation rate of the root. So again, we have time and root elongation rate, and we have these triangles of the overall elongation rate of the root, and you can see it produces very quickly within 5 minutes. But what we see is it's not caused by these red diamonds, and the red diamonds are the length of the elongation zone. So the length of the elongation zone doesn't change. What's changing is the rate at which individual cells are elongating. Okay, and you can track that on a microscope. You can see, you can have a look at the underlying process there. So in other words, within 5 minutes of hitting high aluminium, the elongation rate of the root has decreased, and it's because each individual cell is having trouble elongating. And then you can go and visit the engineers. Okay, and the engineers, in typical style, will treat the root pretty poorly and shove it inside an instron or whatever they happen to have there. Okay, and you can do creep analysis. In other words, you clamp your root in and then you try and pull it apart and you measure the force. And so here we have strain. So these black circles, so you're clamping in and trying to pull it apart, right? And so here's, as you try and pull it apart, what's the strain? So here we have our control, but as soon as you hit your root with aluminium, you can see that there's less ability there for it to elongate. Okay, the root is more rigid. You can't pull the root as far. Okay, and then the final thing I'm presenting here is, yes, Big Z, this is another synchrotron. This is the Italian synchrotron, and that's Alessandra, who runs the machine for us over there, beavering away, changing our sample. And so what we can actually do there is we can actually, for the first time, we're able to measure the aluminium in the plant root. Now that might not sound like much, but it hasn't actually been done before because it's actually very difficult to get a fully quantitative measurement of aluminium distribution in your root. It is actually quite complex. And so here we have 3 parts of the root which we don't need to worry about. This is the outside of the root here. Right? This is outside the root, and the centre of the root is somewhere over here. Right? So this is just the outside of the root. Here's the epidermis, the rhizodermis. Here's the cortex. And the colours represent the aluminium. The brighter the colour, the more aluminium. So where's the aluminium? It's in the outer 5 or 10 micron of the root. Okay, it's not moving into the root. So, each of these images, well, in fact, just an image that size takes 10 hours. So, to get these images, that's probably 3 or 4 days work there. So, was PAX correct? Right? Unfortunately, as always, yes. So, aluminium toxicity, we see these ruptures. We see that these ruptures are caused by an inhibition of elongation. We see that the formation of ruptures is related to the strength with which any of these metals are binding to the root. It's not an aluminium effect, if you like. It's a general common toxic effect caused by anything which is binding strongly to the root, preventing it elongating. We see that aluminium reduces root elongation within 5 minutes, exceedingly quickly. Reduction in the rate of cell elongation is not a reduction in the elongation zone. We see a creep analysis. We show that there's this change in the elasticity of the root, and we show that in low X-ray fluorescence, low energy X-ray fluorescence, we show that this cracking of the root is associated with the accumulation of aluminium in those outer cells. And so it's those outer cells which are inhibited from elongating and tear themselves apart. So, overall summary and conclusions. Soil is critical for survival. Okay? We need soil for our food. It's as simple as that. And so we expect to have 9 billion people on Earth by 2050. How are we going to achieve that? Okay? Soils provide us with $11–12 trillion a year in goods and services. But our expectation, perhaps our hope, is that that's going to increase. We're going to have to double our production per unit of land area. And so we have this grand challenge. But at the same time, we know that our existing agricultural systems, we're losing half of the organic matter from our soils in long-term systems. We're losing half of the nitrogen. We're losing the phosphorus. We're losing the sulfur. We're acidifying our soils, etc., etc. And so how are we going to double production per unit of land area whilst at the same time maintaining and nourishing and nurturing our soils in order to achieve that? And so we have these challenges of, this is what's happening at a global scale, but what's actually underpinning that? How are we going to manage our systems? How are we going to influence our decision making to achieve the outcomes that we need to achieve to feed all of these people? So we lack this fundamental understanding. We need to link the broad-scale approaches that we see with the mechanistic understanding. Soil scientists have an exciting, but no doubt exceedingly difficult task ahead to understand and nurture our soil while meeting the needs of our society. And so I'd just like to thank the many people who have worked with me on this work that I presented tonight over the last decade or thereabouts. And that's UQ, you can see the people listed there, also in Germany with the nano work, and also in Italy with the aluminium work that I showed you. Thank you very much.